Yeah. So hi everyone. So today today's live session 8, where we'll be discussing the last week lectures, week 8. Uh, so after this, on 20th March, we'll have a final session where we'll do a revision of all the classes till now. And if you have any doubts in the whole of the course, you can ask. That's right before the exam. So yeah, I think that will be helpful. 20th March, 6 p.m. to 8 p.m. Okay. Can everyone hear me and can everyone see the screen first? Okay, thanks. Uh, yeah, so here are some important links as before. A link to the live session, the YouTube and the Google Drive where all the presentation are present. Uh, Here are some other important links to download R Studio and the GitHub link for all the codes for the course. You can also go to each of the lectures and then wherever there's a code. So you can basically go here and download the code. So it'll download a zip file or a RAR file and you can extract it to see the code, right? Run the code and the data. Uh, yeah. So we'll start with week 8, analysis of high dimensional data. So a lot of experiments in biology, such as microarray experiments for measuring gene expression, tend to produce a lot of data. So these are like high throughput experiments. So for example, we might have a gene expression study where we are using a microarray to study gene expression. And we have, let's say, 50 cells, 50 different cells, so 50 different samples, and 8,000 different genes. So we are measuring the gene expression across 8,000 different genes. So basically, we'll have a data matrix, and where the rows will be number of samples, and the columns will be number of genes, right? And each of our sample is basically a data point. Right. So if we try to plot this data or like basically if we try to represent this data mathematically, uh, each of our point is a sample. So each of our point is basically one row of this data frame and it is represented by 8,000 genes. So basically there are 8,000 dimensions in which it can vary. So basically our whole data set is now is in a space of 8,000 dimensions. So generally you see this kind of data and high dimensional data and issues when you're typically dealing with high throughput experiments. So uh, the main key problems with high dimensional data is that you cannot basically visualize a data that is anything greater than 3 dimensions. So you can't make plots. You can't see how your data is related, like how your sample points are related to each other. And it's also difficult to extract meaningful and quantitative information between the data points if it's there in a high dimensional space. Okay. So what we do when we have very high dimensional data, we do something called dimensional reduction. So basically, using the original dimensions of the data, we try to combine them in some way to create new dimensions, where the number of new dimensions that we create is very less than the number of original dimensions. So for example, if we wanted to visualize this data set that we have of 50 samples and 8,000 genes. So we'll basically reduce its dimensionality from, let's say, it's 50 into 8,000. So 50 data points across 8,000 dimensions. We'll reduce the dimensions to something like 50 into 2. So we'll now have 2 dimensions. And basically, we again have the 50 sample points. So basically, we are reducing it to 2 because now we can plot it in a two-dimensional axis and visualize the whole data. Great. Uh, So basically, when we are reducing the dimensions, we are projecting the data into a low dimensional space. We can then visualize and analyze the data in the low dimensional space. One important thing to keep in mind when you are reducing the dimension of data is that we always want to reduce the dimensions with the minimum loss of relevant information. So you want to reduce the dimensions from 8,000 genes to, let's say, two arbitrary dimensions, but we want to conserve as much information about all those 8,000 dimensions as possible. So that is one of the main aims of reducing dimensions in high dimensional data. So there are two common dimension reduction methods that are followed in biology. One is called PCA, or principal component analysis, and the other one is TSNE, or T distributed stochastic neighbor uh, section. Just a second. Neighbor embedding, yeah, stochastic neighbor embedding. So basically, we'll first discuss about PCA and then move over to TSNE. So in PCA, what we are doing is we are basically projecting our high dimensional data into a new coordinate system. Okay. And we are choosing a coordinate system such that the new dimensions or the axis that we will have will explain most of the variation that is present in the data. Another important thing to keep in mind is that in PCA, the axes that we follow are always orthogonal to each other. So what does all of this mean basically? So let's say you have a data. Uh, let's say you have some two-dimensional data like this, X and Y. Right. Now we want to do a PCA. So we want to change the axis. So the current axis are X and Y. We want to build two new axes, X prime and Y prime, such that these axes will capture most of the variation of the data. So as you see, if you draw a line here, that will, like, visually, if you inspect, that will basically capture most of the variance in your data, because your data varies significantly, like, along this direction, right? And then once we have one axis, what we do, we find another axis which is orthogonal to this and explains the most variance that is left over. So this X prime axis will explain some of the variance of the data, right? The variance that is particularly in this direction. Then we need a Y prime axis, which will be orthogonal to X prime. So let's say it's like this, and it explains basically some more of the variation in the data, right? So after that, what we do, we basically reproject our data to the new dimensions. So now we'll have X prime and Y prime as our axis, and our data will be like this. Right. So basically, when you are doing a PCA, you map your dimensions, whatever dimensions there are, to some new dimensional space of same number of dimensions that explains most of your variation. Now, what we want to do in PCA, so let's say we were trying to reduce the dimension of this data set from 2D to, let's say, we want a 1D representation. So now what we'll do, since this axis, this X prime axis explains most of the variation in our data, what we can do is basically we can keep this and get rid of this. So basically, if you want to represent this as a one-dimensional data, we'll basically project all the points to the X prime axis here, and we'll basically plot those. So these will be our points now, one-dimensional representation. Right. So this is basically PCA. So uh, we talk a little bit about the math of PCA, but we don't go deeper into how exactly it works and how exactly the equations are solved. And those kinds of things will not come in the exam, so you don't need to worry about all that. You just need to have a general concept of what PCA is and how it happens, right? And how you can use it. So basically, let's say we have data like this, which is a n cross d dimensional matrix, where n is a number of uh, samples and d is a number of variables or the different features, right? So in PCA, what we are trying to do, we are trying to project this data into one vector such that the variance of the projected data is the maximum. So basically here what we did, if you want to mathematically define it, what we are doing is we are trying to basically project this data, the points that we have, into an axis such that maximum of the variation is present in that axis, is captured by that axis, right? So let's say this is our data matrix X, then we have a vector U, which has the dimensions d cross 1 basically. And U is a unit vector, right? So we want to project the data, project X on U. So that is given in matrix terms by XU, which is basically the dot product. So which will be like a matrix of this form. So n into 1, because we are taking the dot product of this. We are multiplying these two. So basically x1 prime, x2 prime, and so on till xn prime. We'll have all the values. Right. So basically, we have reduced the dimensions that we had here, d dimensions, into just one dimension. We have just collapsed everything into one dimension here. And each of the rows here represent where our points fall in that dimension. Okay. Uh, so basically, you can do this for multiple such vectors. So U1, U2, U3, and so on till how many ever you can find, like UD, you can find. And basically, you can project our data into this X U1, X U2, and so on. You project till X UD. So you get projections of the data in the newer dimensions, which are represented by the vectors U1, U2, U3 till UD. Okay. Uh, does anyone have any issues till here? Does anyone have any doubts? Okay. So, yeah. So one thing we also talked about is the covariance matrix. So covariance matrix of a data matrix X. So X is at this matrix here. So covariance of X is defined as 1 by n into X transpose X. So 1 by n, X transpose into X. So n is basically the number of samples that we have, n, and uh, yeah, 1 by n we divide X transpose X. And this covariance matrix is basically a square symmetric matrix. So let's say we are taking covariance of X, which has uh, 2 cross 2 dimensions. So basically x1, x2, y1, y2, something like this, right? So when we take the covariance of this, so the covariance matrix of X will have basically the variance of X here, the variance of Y here, and here it will be the covariance of XY, here it will be the covariance of XY. Okay, the values in the matrix will be this. So this is a square symmetric matrix, and it has various properties. So the first thing is all eigenvalues of F will be non-negative. All eigenvalues of the covariance matrix will be non-negative. All eigenvectors of the covariance matrix will be orthogonal. Okay. And if n is greater than d, and all columns of X are linearly independent. So X is basically n cross d, n number of samples, d number of variables, right? If n is greater than d, so we have more samples than variables. Okay. and all of our columns of X are linearly independent. So all of our variables are linearly independent. Okay, they're not any of them are not linear combinations of each other, right? Then this covariance matrix S will have D number of eigenvectors. So D is the number of variables. So it will have D eigenvectors, and which are represent and which will have eigenvalues lambda 0, lambda 1, lambda 2, which will have eigenvalues lambda 1, lambda 2, lambda 3 till lambda D, which are all non-negative, greater than 0, right? And if n is less than D, so if the number of samples is less than the number of variables we have, then at least one of the uh, eigenvalues will be 0 of the covariance matrix. So these are some of the properties of the covariance matrix, and this is how we define the covariance matrix in linear algebra. Sorry. So why are we talking about the covariance matrix? Because when we define PCA, we define PCA as an optimization problem. So basically what I mean by that is, so we have this thing which is projection of X on U, right? Which is given by this. So the objective of PCA is to maximize the variance of this by finding a suitable value of U. So that is what is represented here, right? So basically the objective of PCA is to maximize the variance of XU by varying the value of U. So we find a U such that the variance of XU is maximized because we want to retain the maximum variance in that unit vector, in that dimension, right? So where does the covariance matrix come in? So basically variance of XU can be decomposed into this. So U transpose covariance matrix into U transpose SU. And if we were to use math and solve this optimization problem, we'll find that the solution is of the form SU equal to lambda U. So basically, the variance of the projected data, the data that we are projecting onto the new axis, will be maximum when the unit vector is the eigenvector of the covariance matrix of the data. Okay, so basically now we have the covariance matrix X, and it will have a lot of, so it will have some eigenvectors and corresponding eigenvalues, right? And these eigenvectors are basically the new axis after PCA. Right. Does anyone have any doubts with this? Okay. So, so S, as I mentioned, is a D cross D matrix. Okay. Assuming that the columns of X are linearly independent and N is greater than D. Okay. So since it is linearly independent and it's a D cross D square matrix, it will have D eigenvalues and eigenvectors. Okay. Now we want to calculate the variance of the projected data for a given value of U. So let's say variance of XU is given as U transpose SU. This again we can write as, so because SU is equal to lambda U, which comes from here, which is our solution, SU equal to lambda U. So we replace it by that. So it becomes U transpose lambda U. And this lambda is a scalar, so we can take it out. So it becomes like take it to the left. So now it becomes lambda U transpose U. And U transpose U, if you multiply, you get the identity matrix. So basically the variance of XU is given by the lambda, the corresponding eigenvalue. So the variance of a given eigenvector is given by the corresponding eigenvalue in PCA. Okay. Now what we do is once we get all the eigenvalues and eigenvectors of the covariance matrix, we arrange it in descending order and we number it as such. So lambda 1 is the largest eigenvalue, lambda 2 is the second largest eigenvalue, so on till lambda d, which is the smallest eigenvalue. Okay. So corresponding to lambda1, the vector u1 is the eigenvector which will contain the highest variance, because lambda is highest and lambda is the variance, right? So lambda 1 is the highest, so u1 explains the highest variance or contains the highest variance of the data, and u1 is called the principal component 1. U2 again will have eigenvalue lambda 2. It is the second highest, it has the second highest, it explains the second highest variance, and it is called principal component 2, and so on till we have principal component D. So we'll have 1 to D, all the principal components. Right. Now we have D principal components now from our data, which are basically D new axis in which we can represent our data. Right. Now what we need to do, so we again have D dimensions, D new axis. We need to discard some axis and just limit ourselves to a few axes. So how do we do that? So basically we plot something called the scree plot. Okay. So we have lambda 1 is greater than lambda 2 and so on till lambda D. Right. Now we convert this into proportions. So basically it will be lambda 1 divided by sum of all the lambda. So sum of all the variance. So variance explained by the first unit vector divided by sum total of the variance. Then variance explained by the second unit vector divided by sum total of the variance. So this gives you a proportion of variance explained. So on the y-axis, we have what is the proportion of variance explained. On the x-axis, we have what are the unit vectors of our PCA, or what are the new dimensions. So basically, we can see if how much of our variance in our data is explained by the first unit vector, how much is explained by the second, how much is explained by the third, and so on. And because we have chosen lambda 1, lambda 2, lambda 3 such that the variance keeps on decreasing, so the lambda keeps on decreasing, so this plot will look like this. So it will keep on decreasing. And after a point, you'll reach an asymptote kind of, or like the value decrease. So like after a point, there'll be like a sharp decrease, or basically like the, let's say for example, this is the first three unit vectors we want to consider in this data, because anything which is this or after this will have very low values. So we can basically discard these unit vectors. So now we can represent our data using just these three unit vectors that we get from PCA. So now we have, from D dimensions, we have reduced it to, let's say, 3 dimensions. Or if you want to just reduce it to 2 dimensions, we take the first two and then we reduce it to 2 dimensions. So that's how PCA works. Okay. Does anyone have any doubts with this? Okay. So, so this is basically what we are doing mathematically when we are doing a PCA. So we have our data matrix, which is of the shape n cross D, where D is our variables and n is our samples, different samples, right? Then we, using PCA, we find optimal unit vectors u1, u2, u3, um. Okay. So basically these principal components. So we select m principal components. Let's say we want to reduce D to 2 or 3. So we'll select 2 or 3 principal components, and we'll have a matrix V, which is basically a these unit vectors are arranged in different columns. These m unit vectors are arranged in the columns. And if we multiply these two, so this is n cross D and this is D cross m. If we do matrix multiplication, we'll get something which has the dimensions n cross m. So let's say here m was 2. So now what we get here is n cross 2. So we have the same number of samples, but now they are represented in 2 dimensions instead of D dimensions, right? So this is represented by the matrix T. So we have the relation X into V is equal to T, where X is basically our centered data. So one important thing for PCA is that before doing PCA, you need to center your data. So centering your data basically means that, let's say you have a column X, which has values X1, X2, xn or something till xn. So you find the mean x bar, and centering is basically the first one becomes x1 minus x bar, second one becomes x2 minus x bar. So xc here, 1 second. Huh. So basically you calculate the mean first, x bar, okay, and then you make the data centered by just subtracting the mean from every observation. Right. So now when you center your data, basically what you're doing, you're setting the mean of your data to 0. So if you calculate the mean of this, okay, then that mean will come out to be 0 when you have a centered data. Right. So basically X is our input matrix, which is a centered data, that we have from our experiment. V is M selected principal component, which is also known as the loading matrix. And T is basically our projected data in how many ever dimensions we want in M dimensions. And this is basically called the scores matrix when we carry out a PCA. Okay. So just to recap, PCA projects higher dimensional data to lower dimensional data while preserving the trends and patterns that are present in the data. Data projected into the new dimensions are projected such that the new dimensions or axes capture most of the variation in the data, right? And these axes are orthogonal to each other, all of them, and these are called as principal components, right? Now, to carry out PCA or to project your data using PCA, what you need to do is you need to get the covariance matrix for your data. You need to get the eigenvectors of the covariance matrix, which are basically the principal components. Then you need to order the eigenvectors based on the eigenvalues. Select the first few eigenvectors, how many ever you want, how much ever dimensions you want to reduce, which have very high eigenvalues. Okay. And then project your data onto those selected eigenvectors or principal components. Okay. So this is it for PCA. Does anyone have any questions? Okay. So we'll move on to how to use PCA in R. So basically, we will be using a package called GG byplot. So you have to install this before running the script. So you can basically load the library DevTools and then use this command install underscore github and then give this address. So it will basically download the package for you. Okay. So when you're running this code, first what you need to do is uncomment these codes and run them. And after that, run the rest of the code. Right. So first we'll read the data. So the data we have is like this. So basically we have 5 columns: group, gene, drug 1, drug 2, drug 3. Basically, this is like testing the effects of different drugs on the gene or something. And groups are basically, let's say these 3 genes, we know they are behaviorally similar, or they are present in one pathway. And let's say these 3 genes are present in another pathway or biologically somehow related. So that's how these groups are assigned. And in each of the drug tests, we are given what is the expression of the gene. Okay. So first, we need to extract the data, the numeric data. So basically, the data is these 3 columns: drug 1, drug 2, drug 3. So what we'll do is ga.data, and then we'll keep this empty because we want to get all the rows of the data. And here we write 3 to 5 because we just run those columns. Right. Then PCA is carried out in R using the function prcom. Okay. So we are basically using the function prcom. we are giving it our data D. Okay, let's just take a look at D. So D is basically now just the last three columns, all the numeric values. Right. So we give this data to PRCom. We write center equal to true and scale equal to true. So what this will do is it will first center our data uh, along the column and then scale it. So this is generally done to, so basically before you use PCA, it's always mandatory to do centering and most of the time scaling of your data. So that's what we are doing. So you don't need to do that separately. You just need to give these arguments to a PRCom function. And it will center and scale your data for you. We are saving this to a variable called d.pca. Now let's take a look at d.pca. So it has a lot of different values: center, scale, X, rotations, SDEF, and all. So we'll take a look at them slowly. So the first thing is we type summary d.pca. So we get a summary of the model. So basically what this gives you is what is the standard deviation of the three principal components. So the first one standard deviation is high, second one standard deviation is high, third one standard deviation is low. So basically this is like how much your data is varying along that axis. So in a way, this is a representation of how much variance your data is capturing along this, how much variance of your data is captured along this axis, right? Then it gives you proportion of variance, which is basically how much of your variance how much of the total variance in your data is captured by PC1, how much of the total variance of your data is captured by PC2, which is 0.37 here, and PC3, which is 0.018. Right. Then cumulative proportion is basically, this is basically the same. This is basically sum of these two. So sum of all the PCs that are before that. So this is sum of these three, which turns out to be 1. Okay. So now we want to make a scree plot. So basically, we have to put the mm, principal components on the x-axis, the principal component number on the x-axis, and the y-axis, it will be proportion of variance explained by the principal component. So how do we calculate proportion of variance explained? So we basically use the d.pca, the PCA. From that, we get the standard deviation. So this is basically standard deviation of all three axes, right? The variance of all the standard deviation of all three axes. So we are basically squaring it to get the variance. So we get the variance and then we divide it by the sum of variance of all the axes, right? So basically we get PV. Now, if we take a look at PV, it's basically what is the proportion of variance explained by all three principal components. We can see this is the same as this. Right. Now we want to plot it. So we plot PV. We give the X label as principal component, the Y label as PVE, the proportion of variance explained. PCH is type of symbol, CEX is the size, color equal to 4, and we provide the Y limits. So we plot this, we get a plot like this. So the first principal component explains almost like 60% of the variance. The second one explains somewhere close to 40%. And the third one explains very low, close to 0, 2%. Right. Now, but this graph is not that clear most of the time. So what we do is we plot the cumulative uh, proportion of variance explained. So basically, we can use the sum function in R to get the cumulative sum of the vector PV. So we get the cumulative sum using the sum function. Then we basically plot. So it's basically the same function, but instead of PV, we have CPV. We run this. We see that principal component 1 alone explains about 60% of the variance. However, if we take principal components 1 and 2, it explains very close to 100% of the data. So it explains somewhere close to, let's say, 95, 97% of the data. right? So basically here what we can do, we can discard our third principal component and just have these two principal components so that we can reduce the dimensions of our data, right? Then again, we had these different variables in the d.pca, the PCA result, the PCA that we carried out, the result of that. So basically, dPCA has something called rotation. So this rotation is basically our loading matrix, right? Which has the dimensions n cross f, right? So contribution of original variables to the principal component. So basically what I mean is, so if I see this, so this is PC1 and this is how much drug 1 affects PC1. So drug 1 is positively related to PC1, basically zero point 0.6, highly positively related. Drug 2 is highly positively related and drug 3 is slightly negatively related. Right. So if drug 3 increases, the value of PC1 decreases. If drug 1 and drug 2 value increases, the value of PC1 should increase. Right. So PC1 is basically a linear combination of these three axes in these proportions. So these are the coefficients of the linear combination. Right. Similarly for PC2 and PC3. Now, the scoring matrix is basically the projection of our data points. It will have the dimensions n cross m, basically. So loading matrix has dimensions d cross m, and this scoring matrix has dimensions n cross m. So basically, if I, so this is given by PCA dollar X. So this X value here is basically our scoring matrix. So now if you take a look at the scoring matrix, it basically gives you where your first point is located, where your second point is located, where your third point is located. So this is gene 1, coordinates of gene 1 in the new transformed axis, coordinates of gene 2 in the new transformed axis, coordinates of gene 3 in the new transformed axis, and so on. Okay. So now we want to plot this whole thing. So we'll be using the ggbiplot library. We'll load it. Then we write P1 is equal to ggbiplot d.pca. We are providing the data frame with the data, basically the PCA data, result of carrying out the PCA. Choices is basically what all different principal components do you want to plot. So we just want principal components 1 and 2, not the third one, right? We are discarding the third one. Ops.scale and var.scale basically scales your data. So we are setting it to 1 because we don't want any scaling or changing in our data. And labels is basically g.data second column, all the rows second column. So what is there? G.data second column is gene. So we are using the gene names as the labels here. Okay. Now we are saving the output of this ggplot to P1. Now we need to print it to plot it. So if we print it and we zoom on it, we see that. So this is our PC1, which explains 60.5% of our variance. This is our PC2, which explains 37.6% of our variance. And these are all the locations of the different points, the genes. Gene 1, gene 2, gene 3, gene 4, gene 5, gene 6. And these arrows are basically, so we'll scale the image so that we can see it properly. So we are basically changing the limits and we are printing again. Okay. So now basically this means this is where drug 1 is, the axis drug 1, this is drug 2, this is drug 3. So basically what this means is if we go to d.pc, d.load, if we go to the loading matrix and we see, so our PC1 is 0.65 into drug 1. So if drug 1 increases, PC1 increases. Drug 2 is also positively correlated, 0.72. So if drug 2 increases, PC1 increases. Drug 3, it's negative correlation, minus 0.2. So if drug 3 decreases, then PC1 right. So basically, if drug 3 decreases, PC1 increases. So these are basically the combinations of this. So again, for PC2, we have drug one, projection of drug one like this. Okay. So basically what I mean is in PC1, if you project all these axes, so this value, the drug 3 where it will fall, it will be minus 0.2. Drug 1 where it will fall, it will be zero point 0.6. Drug 2 where it will fall, it will be zero point 0.7. And again for PC2, it will be something like this. So basically this tells you how our initial axes have been modified, okay, to produce the new principal components. Then what we want to do now is we basically want to plot this data, but we want to color code for groups. So basically we are doing GG biplot again. We provide the PCA data. Choices is basically we just want the first and the second principal axis. Scaling we don't want. Labels we are providing the gene names, G1, G2, G3, G6. And groups basically tells it what to color it by, right? So groups is G data first column. So G data first column is the group. So we want to color it according to the group A or B. And we print it. So this is how the plot looks like. Variance axis is false. So basically it won't draw the axis, the other three axes. So this is how our plot looks like. We have group B, which is this side, gene 4, 5, 6. Group A, which is this side, gene 1, 2, 3. And basically how they like it basically separates them based on makes it easier for us to visualize. So probably G4 is farther away than G1 compared to G5 in the higher dimensional space. That is what PCA tells you. Right, we are basically reducing the dimensions, but you want to keep the variance intact. We want to basically maximize the variance along the principal axis. Okay. Does anyone have any doubts? Okay. So we'll move on. Okay. So that was a sample PCA that was carried out for a very small data set. Now we'll do the same thing with a larger data set. So this is the NCI 60 data set from the ISLR2 package. It contains, we have used this before also in one of the class. It contains gene expression data of 64 cancer cell lines across 6,830 genes. Okay, so basically we have 64 samples and 6831 variables are genes. Right. So now we read the data as G. So we'll take a look at G. It'll take some time to load. There's a big data set. So X1, X2, X3, X4, these are all basically the genes. CNS, CNS, renal, these are basically the cancer cell. The first one is what type of cell it is, and these are the gene expression values. Right. So what we want to do, we want to first extract the labels. So these are the labels. We want to extract it to a separate data frame. So G dot lab will be the labels. So we are taking all the rows and the first column. G dot data is the data without the labels. So we are taking all the rows and we don't want the first column. We want everything else. We run this. Now we see G dot label basically is a character vector which has this first column, and G dot data basically has everything else, x1, x2, x3 till whatever number of genes there are. Okay. Now we want to perform PCA. So we'll use the prcom function to perform PCA. We'll provide it the data, G dot data. We want to center it and scale it. So we'll put center equal to true, scale equal to true, and we save it to G dot PCA. Right. Now we want to make a scree plot to look at the different principal components and how much variance they explain. So again, we'll do G dot PCA. From that, we get the standard deviation square. So basically the variance, and then divide it by the sum of the variance of all the uh, different principal components, right? And then we basically plot it using the plot function. So plot PV, X lab is the label for X axis, Y lab is the label for Y axis. This is the kind of point, the size, and the color. Now we do this and we see. Okay, so if you zoom, we see that basically the initial few principal components explain a lot of the variance. So it goes till point one. So explain not a lot of the variance, but explain higher amounts of variance. And as we increase the number of principal components, the amount of variance explains kind of decreases. So in this case, we might have to take like, let's say 10 or 15 of the data points to capture a lot of the variance. It's slightly difficult to see from this. So what we'll do, we'll plot the cumulative uh, from scree plot, right? So now we use the COMSUM function to get the cumulative values for the PV vector and we plot it. So we see something like this. So basically, we want to select something here, let's say 10 or 20 principal components where we are explaining at least more than 60% of the, which captures at least more than 60% of the variance in the data, right? So but now we just want to visualize the data in 2 dimensions. So for that, we'll use the GGY plot. Okay. So we provided the PCA data frame. Choices are basically what axis we want to plot. We want the first and the second principal components. We don't want to scale it and we don't want to plot the axis, original axis. So we run this, we'll print it. So we get this. So this is basically how our data looks after we reduce the dimensions to just 2 dimensions, PC1 and PC2, which explain slightly less, which explain like this much amount of variance. So 11.4% and 6.8%. Okay. So yeah, so now what we want to do, we want to, so we got just a plot, right? But we want to see how these different types of cancer cells are present in this plot. So how are all the cancer cells from the same type nearby to each other, or they are farther away, or something like that? So for that, what we need to do, what we'll do is we'll basically color these points according to the type of cell they are. Right. So we'll do GG plot. We'll provide the PCA data set. Again, choices is 1 and 2 because we just want the first two principal axis. No scaling. We don't want the variance axis. And in groups, we provide g.lab. So g.lab was basically our labels for the data set. Right. So this in P2 and then print P2. So this is what we get. So for all the different kinds of cancer cell lines, we see how this data is present and segregated. Right. So, for example, the unknowns are here. This is like one group of the similar color. This is one group of the similar color. And so same kind leukemia, I think. Right. And NSCLC is here and so on. So this is one way we can visualize. So basically now what we are doing, so initially we had 6830 genes. So basically our data was in 6800 dimensions and now we reduce it to just 2 dimensions and we are able to plot it using PCL. Okay. Does anyone have any questions? Okay. So we'll move on to TSNE. So before TSNE, we'll talk about stochastic neighbor embedding, SNE. So basically, this is again this is a nonlinear dimension reduction technique where basically the objective is to embed the data from higher dimension to lower dimension so that we preserve the neighborhood identity. So we want to keep points that are closer together in our reduced dimension reduced dimension for, and we want points that are farther in the original higher dimensions to be farther apart in our lower dimensions. Okay, so for this, what we do is we calculate the distance of distance or dissimilarity between uh, any two data points that we have. So basically, let's say you have a lot of data points x1, x2, x3, xj. Okay, X, I, X, J. So basically, we calculate the distance between them. Okay, let's say the Euclidean distance or something. So then what we do using this distance, using this Euclidean distance in higher dimensions, what we'll do is we'll calculate a probabilistic similarity measure. Okay, which is defined as such. So Pij is equal to probable is the probabilistic similarity measure Pij, which is given as Pj given I plus Pi given j divided by 2n. Okay, so Pj given I is again given in this form. Okay, where again we are using the distance here, and we have sigma I, which is a tuning parameter that we can control, right? So basically what we are doing here is we are squaring the distance. We are taking the exponential of the square distance and we are dividing it by 2 sigma square. And here we have it for j and I. And here what we have is across I, everything. So we are basically varying the j, varying the k here. And across all I, we are basically getting the sum of this thing, right? So if like the distance is low, then basically dij square is low, right? Exponent of this is low. So this value is low. And oh, sorry, this is a negative. So this value is high. So if the distance between these 2 points in the higher dimension is low, then this probabilistic similarity measure is high and vice versa. Okay. Is that clear? Does anyone have any doubts? Okay. So this basically this form of representation that we get here, it's basically a normal distribution. This is how a normal distribution looks like. So basically here we are trying to find a probabilistic measure, which is basically a Gaussian. Okay. Then what we do after we find this similarity measure, we randomly arrange all the data points in lower dimension and calculate the Euclidean distance between all pairs of data. Okay. Calculate the Euclidean distance between all the pairs of data. So Eij is basically the Euclidean distance between the data I and j. So let's say we are representing the data in 2 dimensions and we have these two data points I and j. So Eij is basically this distance, Euclidean distance. Right. Then using Eij, similar to how we made this Pij calculation using Dij, what we do is we basically calculate something called Qij. So instead of this Pij, we'll have Q. So basically Qj given I, and this is Qij it will become, and this will again become all P, and this D everywhere will be replaced by Eij. So that's how you calculate Pij, Qij, and here we'll use the value of sigma square as half to make calculation simpler and easier. Okay, so sigma square is a parameter that we change, right? Then what we need to do is we need to compare this Pij and Qij for all the data pairs. And what we want to do is we basically want to find Pij and Qij such that they are very close to each other or they map onto each other easily. Okay. So mathematically, how do we compare Pij and Qij? So basically we calculate L, which is the summation of Pij into log of Pij by Qij. Okay. So what this means basically is if they have basically if Pij and Qij are similar, so if you find 2 points in the space which are similar or which can be mapped to each other, then this will become this whole fraction will become 1. So log of 1 will be 0. So and if we are summing over I and j, so in the ideal case, if all of your Pij can be mapped on into some Qij, what you'll have is basically all of these numbers will be 1 and the log of 1 will be 0 in all the case. So when you take the sum, it will be 0. So in an ideal case, when there is one-to-one pairing for all of these, then you'll get L, which is 0, right? But the ideal case does not happen often. So what we need to do in TSNE, so what we need to do in SNE, stochastic neighbor embedding, is basically we repeatedly rearrange the data on the lower dimension and then calculate this L, and we try to find uh, an arrangement such that the L is minimized. So this is basically our optimization problem. We want to find an arrangement of data in the two dimensions such that the L value that we get here is minimized. Okay. Does anyone have any questions? Okay. So this is basically stochastic neighbor embedding. TSNE is basically T-distributed stochastic neighbor embedding. Okay. So this is also a non-linear dimensionality reduction technique, which basically does the same thing that, which has the same objective, which basically embeds data from higher dimension to lower dimension by preserving the neighborhood identity. Okay. The only difference is that in TSNE, we use a T distribution in place of a normal distribution when we are calculating QI. So in SNE, when we calculated Qij, we use the same formula that we use for Pij. We just replace Dij by Eij, right? While in TSNE, what we do is to calculate Qij, we use this formula, which is basically similar to a T distribution. So why do we want a T distribution? Because when we use a T distribution while doing this, it basically moves the close points closer and it moves the far points farther apart, which is what we want in our visual representation of our data. So basically how it happens is, so let's say this is a distance measure and this is your probability measure, right? Or similarity measure. So, so you have a, right? I'm just doing this to distinguish both the lines. So this is basically a normal distribution. This is basically a T distribution in TSNE. What we are doing when we are mapping from the higher dimensional space to the probability space. So when we are finding out Pij, so let's say we have 2 points xi and xj, which are let's say at a distance of 1 unit. Okay, which are let's say at a distance of 1 unit in the higher dimensional space. So what we'll do is we'll basically, okay, let's say they are at a distance of 2. So what we'll do basically is, mm, so because they are at a distance of 2, when we are calculating the probability value will come somewhere here. Because when we are calculating the probability, we are using the normal distribution, right? And then when we are again, after calculating the probability Pij, we are transforming, like changing the value to Qij and then using it to transform to our uh, embedding space. So basically what happens is Qij value is taken as this, and then we use the T distribution to basically map the data to lower space, lower dimensional space. So what happens is basically using the T distribution, the distance you get is let's say 1.5, which is smaller than 2. So basically, if the points are close apart, if they're just 2 units farther away, then they'll be closer apart in the TSNE transformed place, right? Now, what happens when they are farther? So let's say the point is somewhere at 3.5. So let's say the distance between xi and xj is somewhere 3.5 or something, right? So we'll use this normal distribution to first get the probability. We'll get a probability of, let's say, this. Then we extend it to the t distribution and we go back to the distance. So if the distance is 3.5, now it becomes 5 point something. So if so basically things that are closer to each other will come slightly more closer, and things that are farther in the higher dimension will become more farther in the lower dimension space. So this basically helps us in our visualization and dimension reduction. That's why we use a t distribution in TSI. Does anyone have any doubts up till here? Okay. So as mentioned, the sigma value that was used, the uh, sigma here and basically the sigma here, the sigma in also in TSNE while we are calculating Pij, the sigma is controlled by a tuning parameter called the perplexity. Okay. So in a way, this perplexity basically balances the local and global aspects of the data. Okay. So if you have a lower perplexity value, it focuses more on the local aspects of the data. And if you have a higher value, it basically uh, drives the focus more towards the global aspects of the data or the relationships between the data in the higher dimensions. Right. Ideally, the perplexity should be always smaller than the number of data points, and the range suggested range is 5 to 50. If you change the value of your perplexity, your plots will turn out to be very different, and it might be difficult to. Uh, so you might not get actual representation of your data. So this is something that you need to be careful with when you're choosing a value for perplexity. Okay. The other thing is when we are carrying out analysis using TSNE, uh, so you might get, for example, let's say you have some data like this. Okay. So you have basically two groups in your data. This is a very varying group, and this is the slightly less varying group, very like concentrated group. But after you do a TSNE, what happens is, let's say your points get mapped into some other space where they look like this. So the upper and lower points, both of them look. So now again you have two clusters, which are basically these clusters, but the variance has changed. Right. So basically, the size of your cluster, how big it is, how small it is, does not matter. So if you take, so let's say this is for perplexity is equal to 25. Now let's say you take P is equal to 5. What might happen is you might get a very different plot. You might get all your points like this. Right. So you should be careful that you don't infer anything about the original cluster sizes or the variance within cluster based on the variance that you get here. This is just a visual technique, right? To like or the data reduction technique, not a clustering technique, right? The other thing is distances between different clusters that you may get after doing TSNE or PCA may not mean anything. Right. Because you're constraining your data from such high dimensions to low dimensions, some way or the other, there is a loss of information. And the distance that you get from the clusters, between the clusters or any clustering that you see or the size of cluster that you see, are basically not things that you can analyze and not things that are basically um, like, they're not actually like clusters or they might not mean mathematically anything. Okay. So basically, just to recap, TSNE is a tool for visualization of high dimensional data, mostly used in biology. It embeds data into a lower dimension while preserving the local information at the higher dimensions. Okay, so if points that are closer together in the higher dimension will be closer together in the lower dimension, and points that are farther apart in the higher dimension will be more farther apart in the lower dimension. Right. It uses a probabilistic measure of distance with dissimilarity using normal distribution and t distributions. Okay. SNE, stochastic neighbor embedding, basically uses both normal distributions uh, in both cases. So when we are doing the lower dimension transformation and the higher dimension transformation, whereas TSNE uses a T distribution while embedding the data in the lower dimension, but uses a normal distribution on the other side, on the higher dimension. Okay. So this T distribution, what it does is basically it moves closer points together and pushes farther points farther away. Okay. Uh, Another thing is key tuning parameter for TSNE is perplexity, and based on your value of perplexity, your uh, the visualization that you'll get from your data will be very different. So keep that in mind and try to use a good value for perplexity within the suggested range of 5 to 50. Okay. Does anyone have any doubts regarding TSNE? Okay. So we'll move further to uh, using TSNE in R. So basically, this code uses these two packages, ITSNE and ggplot. Okay. So we are first basically install this package. If you have not, you can go to tools, install packages, and then type ITSNE and install. So first we load these libraries, ITSNE and ggplot2. Okay. Then we read the data. So it's a big data frame, so it takes time. So basically what we have are basically uh, gene expression. So these are the different samples, and these are basically the different genes and the count. Okay, so we have count data. Then again, we have some metadata also. So what is the metadata? So, okay, so in the data column, we have basically 16384 columns or 168 genes and 366 samples. Okay. And here we basically, in the metadata, we have for each of the samples, what is like the different things such as the age, brain area, and the cell type and everything. So what we need to focus on is the cell type here. So there are four types of cell: IPC, RG, neuron, and interneuron. So RG is basically radial glial cells, neuron is neuron, IPC is intermediate progenitor cell, and interneurons. Okay. So we want to see basically how we want to basically visualize this data. Okay. So first we'll store the inferred cell type, this all this data, the label data for all this data points that we have into a new vector called cell type. So cell type is meta dollar inferred cell type. And this. Okay. Then basically what we are doing is we are setting the seed of a random number generator. So basically, when you want to carry out analysis that is reproducible, you basically set a seed so that R will generate the same random numbers every time you run the code, and you will have the same model. You won't have any variation. Okay, it's a good practice to do that when you're working. Okay, so we'll use the function RTSNE, which has a lot of arguments. We'll discuss some of them. So basically, perplexity is the argument. This controls our sigma value. The suggested range is 5 to 50. DIMS is basically what is the output dimensionality. The default value is 2. If you don't give a value here, it will produce two-dimensional output. PCA, default is true. PCA is basically before doing the TSNE, it basically carries out a PCA. Okay, if you provide true, if you provide it false, it won't carry out a PCA. Check duplicates will check for duplicates in the data set. Okay, ETA is basically the learning rate or how quickly the model changes. So the default value is 200. It's suggested to keep it from 10 to 1000. Okay. Theta is basically the speed or accuracy trade-off. So if you want to increase the speed of your computation but you can sacrifice a little bit of your accuracy, then you can use a higher value. If you set it to 0, then it will give you the exact values, but it might take a longer time. And so if you can increase the value more to like get faster results, that might not be highly accurate. Okay. Max iter basically gives you the number of iterations. Default is 1000. So we are doing our TSNE and storing it into a variable, TSNE results. So basically our TSNE data is data that we read before, the count data. Perplexity we are giving 30. ETA, the learning rate is 1000. And max iter is the maximum iterations. We give it 5000. And we run this. So it'll take a little bit of time. Okay. To run. Then what we'll do is basically we'll convert it into a data frame. Okay. And basically visualize the cell types in 2 dimensions. So we got it. So TSNE results, it has a lot of different things, right? So we'll convert it to first into a data frame. So y is equal to as dot data dot frame TSNE results dollar y. So y is basically our the thing that we are interested in, the reduction. So if I see y, so y has two columns, v1 and v2, which represent basically the lower dimensions. Since we didn't provide a DIM here, by default it converts our data to, projects our data to 2 dimensions. Okay, so now what we want to do, we want to visualize these. So we'll be using ggplot to plot it. So ggplot, y is the data set. Okay. In aesthetics, we give x is v1. So x is v1, y is v2. Color is basically what color we want it to be. Color we want it to be according to the cell type. So basically the cell type, inferred cell type, this thing. So we want to color it according to the cell type. Then, so we have the data, we have the aesthetics. Now we provide the geometry. So geom underscore point, we want point data. Labs is basically the labels, x-axis label, y-axis label, label for the legend, the theme, which is classic. Now we run this and we see that this is how our data looks like. So basically there are a lot of these RG cells here, a lot of these interneuron cells here, and the other ones are quite mixed here and there. Interneurons and neurons are mixed here and there. IPC is also all over the place. Right. So this is one way to visualize the data where we are basically coloring the data according to the cell type. Right. What we can do also is, sorry. So we can basically color the data according to expression of a certain gene also. So how to do that? So basically, again, we'll use ggplot y aesthetics x equal to v1, y equal to v2 because we're plotting on the lower dimensions, the 2 dimensions that we lowered it to. Color is basically data dollar neuro d6. So data dollar neuro d6. It has a lot of columns. Okay. So neuro d6 is basically one of the columns of the data, which has numerical values for each of the samples. So we'll use that to color. Then geom point, again labels. Then we will use a gradient of color, which goes from gray to high. So the lower value will be represented by gray and the higher values will be represented by red. Now we run this, you see. So here we see that for all our gene, now we see in 2 dimensions where our genes are and what is the expression of this gene in these different points, in these different uh, samples. Okay. So if it's red, it's higher. If it's gray, it's lower. Okay. Does anyone have any doubts regarding this? Okay, great. Okay. So in the class, sir also talked about diffusion maps as another technique for dimension reduction. So diffusion map is also a nonlinear dimensionality reduction technique where it integrates local information to generate a global description of the data, and it is more robust to noise. So it basically follows the structure of the data to, and it tries to like keep local relationships in the data prominent when it's plotting the data. Okay. So basically, we have to measure similarity between uh, various different data points. So we do this by following the structure that is present within the data. We do this using a model called diffusion over data points. So basically, let's say you have two data points, x1, x2, x3, and x4 here, right? So how we measure the distance is by using diffusion. So basically, let's imagine that I have all these data plotted somewhere or in the higher dimension. And if I put a marble here on x1, okay, and give it time, where will it go? Or where all will it move? So basically, will it go here, then here, or from here, then here, and it can also go to itself and so on. So basically, this thing will transition from one data point to the other and so on. Now, the transition probability between two data points is given by this. So for PAB is proportional to basically e raised to the power a minus b whole square by alpha. So a minus b whole square is some other Euclidean distance between them, right? So if the distance is less, then it's more likely that the transition will happen from x1 to x3. For example, in this case, it's more likely the transition will happen between x1 and x2 than between x1 and x4, just because they are farther apart, right, in the higher dimensional space. So what we do basically is, so PAB is basically the transition probability from two data points A to B. What we need to do is for our data, whole data frame, we need to generate a transition probability matrix. So P is our transition probability matrix, which will have like P11, P12, right? P P12, sorry, P21, P22, and like so on. Right. So this is basically what is our transition probability from 1 to 1. What is the transition probability from 2 to 1? What is the transition probability from 1 to 2? So how likely is our marble going from 1 to 2? How likely is our marble to go from 2 to 1? How likely is it to go from 1 to 1 in one step? Right. And so we first calculate this transition probability matrix for our data. And this matrix will have some properties, which is one of the eigenvalues of this transition matrix will always be 1, and the rest of the eigenvalues will be either less than or equal to 1. Okay. And the other interesting property is that, so if you basically multiply this matrix by itself, let's say you find p square or p cube or p to the power t or something. So the values that this will contain, pt or p square or p3, will basically be the sum of probabilities of all paths of length t between any two given data points. Right. So, for example, uh, this pt will have elements p11t, p21t, and so on, and p12t, p22t, and so on. Right. So each of these elements, this one is basically what is the sum of probability of all the paths of length t from 1 to 1. So let's say we are moving from x1 here, and let's say t is 2. So we go from x1, so let's say plot it again, x1, x2, x4, and x3, right? So we transition from x1 to x2, then in one step, in the second step, we go back to x1. So this is basically in two steps, we are going from x1 to x1. What is another way to do that? We go from x1 to x3, they go back to x1. We go from x1 to x4, then go back to x1. So each of these will have some probability. The summation of all these probabilities is this value. So it basically tells you some probability of all the paths of length t. So in this case, t was 2. So if I have t equal to 3, then x1 can go to x2, it can go to x4, then come to x1. So that might be one of the paths. Another will be x1 to x2, then x3, then x1. That will be another, and so on. So we'll have the sum of all these uh, probability of all these paths in the PT matrix. Okay. So diffusion distance is defined as dt between x1 and xi and xj square is given as summation of k pik t minus pjk t whole square. So basically, this is the Euclidean distance. Uh, yeah, this is not the Euclidean distance. This is basically the distance between these two. Pij. This is the sum of the probabilities. Sorry, and this is also the sum of the other probability. So path length from xi to xj. So this is what is the probability of path length from I to k. What is the probability of path length from j to k? You take the difference, you square it, and you sum it for all the different k values that you have in your data, for all the different data points. Okay. And using some maths, you can show that yi minus yj whole square is equal to, so basically what is this? So before we come to this, so PT matrix is something, will look something like this, right? So P matrix, you understand, P matrix is basically has the matrix of all the transition probabilities. PT is basically that P matrix raised to the power T. So P matrix multiplied by itself T times, right? So this is the PT matrix, and each of these rows is basically sample, right? So each of these rows is a different sample. So it can be shown that if we take these two samples and take the Euclidean distance between them, then that is the same as this diffusion distance, right? So basically this now becomes the vector for the ith data point, and this becomes the vector for the jth data point. And now we'll be able to represent our data in n-dimensional space using this yi, yj, using this basically yi. So we represent all of this in n-dimensional space because this has dimensions of n, and we can represent it in n-dimensional space where n is less than the initial dimensions of the data. Right. The additional benefit of doing this is that this preserves the diffusion distance between the data points. So when you are plotting in these n dimensions, okay, in these n dimensions, you are plotting, and let's say you have 2 points, the Euclidean distance between them will be mathematically equal to the diffusion distance between them in a higher dimension. So basically, you are representing the data accurately in lower dimensions based on the distance between data points, right? So how we can basically, so we understand that basically we can represent our data in this form, yi and yj, but we can also then what we do is, so let's say the ith sample, uh, we represent using the form here from obtained from the PT matrix. Okay, T is something that we choose. So we have in n dimensions, the point yi is given as this value, right? We can again make it to less dimensions by basically doing so. So we basically calculate the eigenvalues and eigenvectors of the transition matrix P. And as before, as we know before, the first one of the eigenvalues of this will be always be 1, and the rest of the values will be less than or equal to 1. Okay. So if we make it into eigenvectors, so let's say we have psi 1, psi 0, psi 1, psi 2, and so on till psi n. And correspondingly, we have basically lambda 0, lambda 1, lambda 2, so on till lambda n. Okay. So basically, we can discard this first one because this lambda 0 will be equal to 1. We'll choose the one which has the value 1. And this lambda 0, this psi 0 is basically a constant. So this vector is basically constant values, and that is something we won't need. So we'll get rid of that. So now we can represent our data in n minus 1 dimensions using this formula. So basically PT I1 we have converted into lambda 1 T psi 1 I. So basically how this comes is PT I1 will be basically lambda 1 T psi 1 I. Okay. And so on for all the different, and we leave one of them. So we'll have n minus 1 values here. Okay. Now we can further reduce the dimensions to lower dimension by we'll first arrange these eigenvalues so that they are in decreasing order, lambda 1, lambda 2, lambda 3, and so on. So we'll keep the eigenvalues just as with PCA. We'll keep the eigenvalues that are high, and we'll discard all the eigenvalues and eigenvectors that are low, that have low eigenvalues. So let's say we select these three, then we can represent our data in 3 dimensions from the original n dimensions, from the original d dimensions, right? So now we just have these three, which represent our data in these 3 dimensions. And this will be following the diffusion distance. So basically the distance between these points will be approximately equal to the diffusion distance between them in higher dimensions. Okay, so this is again a data visualization technique. So this is an example where these people have used single cell gene expression data having 48 genes and 429 cells. So basically we have 429 cross 48 data in this format. So this is our initial dimension, 48. Now they have reduced it to 3 dimensions here using diffusion distance and diffusion map. And then they have again reduced it to 2 dimensions using these two components. Okay, and the color is according to the cell types. Okay. So yeah, that's it for diffusion maps, TSNE and PCA. Does anyone have any questions regarding the lectures for this week? Okay. So we'll move on to some practice questions. So the first question is, we performed PCA on 5-dimensional data. Okay. Uh, the eigenvalues of the covariance matrix are given as 1, 4.5, 0.6, 0.01, and 2. Calculate the fraction of the total variation in data explained or represented by the third principal component. So first, we have the eigenvalues. First, what we'll do, we'll arrange the eigenvalues in descending order. So 4.5, 2, 1, 0.6, 0.01. Okay, so we have this. So after you arrange it in descending order, this will be our PC1, PC2, PC3, PC4, and PC5. Okay. So what we want is calculate the fraction of total variation in data explained by the third principal component. So the variation explained by the principal component is basically given as the eigenvalue, right? So as we saw before, when we are talking about PCA, the variance explained by a given eigenvector is given by the corresponding eigenvalue. Okay. So basically, we need to find fraction of the variation. So it will be for PC3, the variation is 1 divided by fraction. So we'll take all the sum of all the variation. 4.5 plus 2 plus 1 plus 0.6 plus 0.01. So the answer is basically this 1 divided by, so variance explained by PC3 divided by total variance. Right. So it's 1 divided by 8.11, which is equal to approximately 0.123. So the answer here will be 0.123. Does anyone have any doubts? Did everyone understand? Okay. The next question is, which of the following statements is wrong? A covariance matrix is a diagonal matrix. That's basically wrong because a covariance matrix is not diagonal. It has variance on the diagonal and covariance on the other places. All eigenvalues of a covariance matrix are non-negative. This is true. We discussed this. All eigenvectors of a covariance matrix are orthogonal. This is again true. A covariance matrix is a square matrix. This is also true because a covariance matrix is given by, let's say, x transpose x, 1 by n. So this is basically a square symmetric matrix. Hmm. So the answer here will be A. This is the wrong statement. A covariance matrix is a diagonal matrix. Okay. The third question is, we perform PC on 5-dimensional data. The eigenvalues of the covariance matrix are 1, 0.6, 2, 0.1, 0.3. We want to calculate the cumulative proportion of variance explained for the principal components. Which of the following R function will be used to calculate the cumulative value? So the answer here is sum, which is the cumulative sum function. Scale is basically used to scale the data. Center is used to center data. Sum is basically the sum. This is basically the cumulative sum. Okay. The fourth question is, which of the following is a tuning parameter of TSNE? Perplexity, t-value, number of clusters, eigenvalue. So eigenvalue is not something you can change. Number of clusters is not something we are looking at. We are not clustering. T-value is not something you can change. Perplexity is a tuning parameter in TSNE. It controls the sigma parameter that is used. Okay. The next question is, which of the following algorithm is usually recommended to be performed before TSNE on data, the very large dimension? So generally before you do TSNE, and if you have a very large number of dimensions, you do a PCA first to reduce the dimensions and then carry out TSNE. So this is a simple question, straightforward answer. Okay, the answer is PCA. Next question is, in a single cell RNA-seq experiment, we measured the expression of 12,000 genes in 40,000 cells. Each of these cells is a data point in the gene expression space. What is the dimension of the gene expression space in this experiment? Okay, so we measured the expression of 12,000 genes in 40,000 cells. So our data matrix will be like this. So we'll basically have cells here, which are our samples. So this will be 40,000 rows. And we'll have genes here, which will be our variables. So we'll have 12,000 genes. So our dimensions would be 40,000 into 12,000. So the a dimension of the gene expression space here will be 12,000 dimensions, because our samples are all our 40,000 points, data points. So these will be our data points, and this will be our samples, and this will be our dimensions per gene. Is this okay? Does anyone have any questions? So which of the following statements is wrong for PCA? PCA is a dimension reduction technique. True. PCA is sometimes used before performing TSNE. True. PCA is a technique for data clustering. False. In PCA, we maximize the variance of data projected on a unit vector by choosing a suitable unit vector. That is basically the principle of PCA. So this is true. The answer here is C, which is false. PCA is a technique for data clustering. Okay. So that's it for the practice questions. Does anyone have any doubts regarding this week or any other week's lectures or our sessions? Okay. So I'll be here for 5-10 more minutes. If anyone has any questions uh, regarding this week's lecture or any other lecture before that, any other week's lecture, please feel free to ask. If you have any doubts with coding, if you have any doubts with R or any issues with R, please feel free to ask. And if you have any specific problem to discuss or anything, please let me know. Another thing is, so the last class is supposed to be on 20th March, which is right before your exams. So 20th March, Wednesday, we'll have class from 6 to 8 p.m. So I was thinking I'll go over the lectures one by one. Just quickly, I'll discuss the key points. And after that, maybe we can discuss a few questions, or if you have any doubts, you guys can ask. So I just wanted to ask, what would you guys prefer? Do you have any specific part of the course that you want me to focus on? Or do you have any uh, particular questions that you have or set of things that you would like discuss in the next class? Please let me know. Uh, that's the main thing. And yeah, that's it. We are done with the whole course. Good job sticking till here, going through all the lectures and the live sessions. If anyone has any uh, questions regarding anything, feel free to ask right now. Otherwise, you can ask it on the discussion forum, which is regularly updated, and people answer there very regularly. So yeah, I'll just wait if anyone has any questions. Otherwise, the class is done for today. Thank you for attending. Thanks. Which Euclidean distance? Today's lecture or before? Can you just unmute yourself and tell me or type it? Virami, which one do you want? Topic about Euclidean distance. 1 second. I'll just check the previous. Okay, I'll just share my screen. So, are you talking about this part? Distance measures for clustering the Euclidean distance?
Yes, sir.
Okay. So, basically, let's say you have 2 points. Okay. X1, two, Y1, Y2. Okay. So Euclidean distance between these 2 points is defined as xi minus yi whole square summation root over. Okay. So xi minus yi basically means you have these 2 points, right? So you have these coordinates x1, x2 and y1, y2. So what you are doing in Euclidean distance is you are taking this difference, squaring it, taking this difference, squaring it, adding both of them and taking the square root of them. Okay. So basically what you are doing here is, okay, I'll just create a new slide, project it. Can you see this? It's visible, right? Hello?
Yes, sir.
Yeah. Okay. So let's say you have this 2 point, x1, x2 and y1, y2. Okay. So basically what this means is this is x1, this is x2, this is y1, and this is y2. Right. Now the Euclidean distance is basically this distance. Right. So how can you calculate this distance? So we'll use Pythagoras theorem basically. So let's say we made a triangle here 90-degree. Okay. Then the hypotenuse is basically the distance that we want. So this is the Euclidean distance. Right. So how can we get the hypotenuse? It will basically be the square of, let's say, A and B, the square of this plus the square of this. So DE square will basically be equal to A square plus B square. So DE will basically be equal to root over of A square plus B square, right? And A is what here basically? A is basically x1 minus y1, x1 minus y1 whole square. B is x2 minus y2 or y2 minus x1, y2 minus x2 whole square, whatever. So since we are taking a square, it's okay. It doesn't matter. Right. So this is basically what is meant by Euclidean distance when we write summation of xi minus yi whole square root over. So basically for all of the corresponding elements, We take the difference, we square it, add it, and take the root. Is this clear?
Yes, sir.
Okay. Yeah. So basically, it carries over to multiple dimensions as well. So for example, this is basically two-dimensional data. You have two dimensions, right? 2 points. Let's say you had x1, x2, x3 till xn, and y1, y2, y3 till yn. So this is your first point, this is your second point, right? In this case. So in this case, again, for to take the Euclidean distance, you can again do the same thing. You subtract this x1 minus y1 whole square, x2 minus y2 whole square, and so on. You add all of them and take the square root. So you can do this for n-dimensional data. And Euclidean distance basically means the shortest distance between these two lines, that you shortest. So basically the straight line passing through them. What is the distance between both of these points? Okay. Is that clear?
Yes, sir.
Yeah, thanks. Do you have any other
Thank
doubts or questions? Okay, so we'll stop here. Uh, Thank you for attending. We'll meet next Wednesday at 6 p.m. for the final class. Bye.
thank you.
Yeah, bye. Thanks.